Question number 1. Plants absorb light for photosynthesis. 1.1. It is an AO1 question. This question is from photosynthesis and photosynthetic reaction. Which is the equation of photosynthesis? It is an AO1 question. The student must have the knowledge about photosynthesis. This is a chemical process which happens in plants to make glucose. The plants need carbon dioxide from the air and water from the soil to make glucose, and oxygen is produced as byproduct. The correct answer is the plants need carbon dioxide from the air, water from the soil, glucose is formed, and oxygen is released into the air. In the apparatus, we have lamp which produces light. We have pondweed in the water which is doing photosynthesis because light is falling and oxygen gas is produced here. A student investigated the effect of light intensity on the rate of photosynthesis. Figure number 1 shows the apparatus. If you see the apparatus, the lamp is present, the plant is present at a distance of 10 centimeter, which can be moved towards right side at about 20 centimeter, 30 centimeter, 40 centimeter, and 50 centimeter away from the lamp. The method the students have used for this process is, or for this investigation is, set up the apparatus, which is shown here in figure number 1. Second point is, place the pondweed 10 centimeter away from the lamp. Switch on the lamp. Number 4, record the number of bubbles of the gas produced in 5 minutes. Point number 5, repeat steps number 2 to 4 with the pondweed at different distances from the lamp. 1.2 is an year 1 question which is associated with the practical activity number 5. What was the independent variable in the investigation? The student must know the independent variable. The independent variable, the variable you can change. Here you can change the distance of the pondweed from the lamp. The length of piece of pondweed is a control variable. You can't change this thing. The number of bubbles of the gas produced, it depends upon the distance of the plant from the lamp. Time taken to collect the gas is fixed, that is 5 minutes, so it is also a control variable. The distance of the pondweed from the lamp is an independent variable. The lamp gets warm when it is on for the longer time. This causes the temperature of the water to increase. 1.3 is an EO3 question from the rate of photosynthesis, and it is also associated with the practical activity number 5. Explain how an increase in temperature would affect the result of this investigation. As the temperature will increase, the rate of photosynthesis will increase. As the result of that, more bubbles will be formed and the volume of the gas will increase. The reason is the enzymes will work faster at the high temperature. Let's see the model answer. The model answer is the enzyme will work faster means increasing the rate of photosynthesis. So more bubbles of the gas mean oxygen gas will be formed. The volume of the gas will increase. One point four is again an AO3 question and related to rate of photosynthesis, is also associated with the practical activity number 5. Suggest one way the investigation could be improved so the temperature of the water does not increase. Because if the temperature of the water will increase, the rate of photosynthesis will increase and more bubbles will be formed. To control the temperature of the water, it is very important that students should use LED lights because they don't emit thermal radiation. Therefore, the temperature of water will not increase. Let's see the model answer. The model answer is put the tube in the water tank. The student can use LED lights because LED lights they don't emit thermal radiations. Use the water tank between the lamp and the water tube. Remember, along with the light rays, the infrared radiations are coming which increase the temperature of water. 1.5 is an AO3 question from the same topic, the rate of photosynthesis associated with the practical activity number 5. Suggest two improvements to the investigation so the result would be more valid. Valid means that the results are fair. Do not refer to controlling the temperature of water in your answer. There are two ways the student can improve the investigation. Number one, measure the volume of gas collected in the gas range or measuring cylinder. Repeat the experiment and calculate the mean volume of the gas. The student needs to use the same lamp as a control variable, same size of pond weed in each experiment, and use same concentration of carbon dioxide in the water. Then the results or the investigation will be considered as valid. Let's see the model answer. Measure the volume of the gas collected in a gas syringe, which can give you more accurate reading, or use the measuring cylinder to collect the gas. Repeat the experiment and calculate the mean volume of the gas. Use same lamp, use same size of pond weed in each experiment, use same concentration of carbon dioxide in the water. All of these three are considered as control variables. Table number 1 shows the results. The distance of pond weed from the lamp in centimeters. The distance is increasing. Number of bubbles of the gas produced in 5 minutes. Number of bubbles in 5 minutes are also shown in the column. 1.6 is an AO2 question associated with the practical activity number 5. And this topic is from rate of photosynthesis. Calculate the rate of photosynthesis when the pond weed was 40 cm from the lamp. Give the rate of photosynthesis as the number of bubbles of the gas produced per minute. The important information is already highlighted. For 40 cm, the number of bubbles are produced, they are 16. These number of bubbles are produced in 5 minutes. We need to find the number of bubbles produced in 1 minute. Therefore, what we need to do, we need to divide the number of bubbles produced by the total time taken. That is approximately equal to 3.2. Or the student can say approximately equal to 3 bubbles are produced in 1 minute. Let's see the model answer. The number of bubbles in 5 minutes are 16. The number of bubbles produced in 1 minute means 16 divided by 5. You will get about 3.2 bubbles, which are actually the 3 bubbles are produced in 1 minute. 1.7 is an AO3 question associated with the practical activity number 5 in the combined science. Give one conclusion that can be made from table number 1. And this question is from rate of photosynthesis. If we go back to the table, we can see that as the distance is increasing, the number of bubbles produced by the pond weed is reducing. The reason is, as the distance is increasing, the light intensity is decreasing and it affects the rate of photosynthesis. If less photosynthesis will happen, therefore less oxygen will be formed or less bubbles are formed. Let's see the model answer. As the distance of the pondweed is increasing, the intensity of light on the pondweed is decreasing. Therefore, the rate of photosynthesis is decreasing, or less bubbles are formed. 1.8. Plot the data from table number 1 on figure number 2. To facilitate the student, I use a table here. The table number 1. The distance of pondweed from the lamp in centimeter is along x-axis, whereas the number of bubbles produced in 5 minutes is along y-axis. Students need to plot these values on the table. The model answer is already given here. At 10 centimeter, the number of bubbles are 120. At 20 centimeter, the number of bubbles are 56. So this is 50, 52, 54, 56. At 30 centimeter, it is 31. At 30 centimeter, it is 31, which is, this is 32, this is 30, between them is 31. At 40 centimeter, it is 16. At 50 centimeter, it is 10. So we need to draw a line of best fit. A lot of the students, they usually make a mistake by drawing a straight line from this point to this point, and they are unable to score full marks. The line of best fit in this case is a curved line passing through these points. One point nine is an AO3 question from the rate of photosynthesis, and it is required practical activity number 5. Predict the number of bubbles that would be produced in 5 minutes if the pond weed was 60 centimeter from the lamp. If we produce our graph further beyond, which I already have done it, at 60 centimeter, we can see the trend. At 60 centimeter away, the graph is passing from this area, and the number of the bubbles are approximately between 6 to 8. So the number of bubbles produced at a distance of 60 centimeter from the lamp is 6 or 7 or 8. The student can use the model answer between 6 to 7 or 6 to 8. So number of bubbles produced in 5 minutes at the distance of 60 centimeter is approximately equal to 6 or 7 or 8 bubbles are produced in 5 minutes. The student can use the answer between 6 to 8. Question number 2. Describe how to test a sample of food for protein, starch, and sugar. Give the colors that would be seen if the food sample contained protein, starch, and sugar. It is an AO1 question, and this question is from required practical activity number 3, and this topic is from the human digestive system. The initial steps are almost same: to test the protein, to test the presence of starch, and to test the presence of sugar. The student needs to mention the change of color during the process or during the investigation. Let's see the model answer. The protein test. Grind sample for protein test and transfer 5 cm cube of the sample in the test tube and add 2 cm cube of biuret solution in the test tube. Shake the solution gently. If the food contains protein, the student will observe the change of color, and if the color of the solution changes from blue to purple or the black, it indicates that the protein was present in the food sample. Student will get 1 mark here to mentioning the process and 1 mark for mentioning the color. Our second test is for the test of starch. We will repeat the same process, grind the food sample for starch test and transfer 5 cm cube of the sample in the test tube and add a few drops of iodine solution. Student will get 1 mark of writing the clear information which is given here in initial step. Shake gently. If the food sample contains the starch, the color of the solution changes from orange to blue or black color. Student will get 1 mark for writing this process and 1 mark for mentioning the color. The change of color is very important to get full 6 marks. Test for sugar. Grind the food containing sugar and mix the water. Add 5 cm cube of the sample in the test tube and add a few drops of Benedict solution in the test tube. A few means from 5 to 10 drops of Benedict solution. Extra information is keep the test tube in the water bath having the temperature of 70 degrees Celsius. If the sugar sample contains sugar, the color of the solution changes from blue to green, then yellow, or even brick red color. The student will get 1 mark for mentioning the change in color and 1 mark of writing the process. Question number 3. Fermentation in yeast is used in the manufacture of bread and alcoholic drinks. The equation of fermentation is given below. The glucose is converted into ethanol and carbon dioxide as a result of fermentation. It is an A1 question, and this question is from the topic of aerobic and anaerobic respiration. Fermentation is an exothermic reaction. What does exothermic means? Exothermic reaction means that heat is produced and it is released into the surrounding. The student must know the definition of exothermic or endothermic reactions. A chemical reaction which releases heat to the surroundings is called an exothermic reaction. A student investigated the rate of temperature on fermentation in yeast. The figure number 3 shows apparatus. The gas range is used here to measure the volume of gas produced as the result of fermentation, which is carbon dioxide. Thermometer will help to show the change in temperature. Water bath will help to maintain the temperature of the environment. This is the yeast solution in which the yeast and the sugar solution is present, and the layer of oil is on the top of that. The student is going to use different steps to investigate this process. The important steps of the investigation are: mix yeast with the sugar solution in the flask, pour a layer of oil over the surface of mixture, put the flask in water at 2 degrees Celsius and leave for 20 minutes. Attach a gas syringe. Record the volume of gas collected every 5 minutes for 30 minutes. After 30 minutes, move the flask to a water bath at 35 degrees. Continue to record the volume of gas collected every 5 minutes. Three point two is an AO3 question. It is an investigation based question, and this question is from the same topic of aerobic and anaerobic respiration. The question is: suggest why a layer of oil was needed on the surface of the mixture. The student must remember that it is a fermentation process. During the fermentation process, the glucose is converted into ethanol and carbon dioxide in the absence of oxygen. It is actually an anaerobic respiration process. Therefore, the layer of oil is used to keep the oxygen away from this process. Let's see the model answer. The model answer is: the oil is used to keep the oxygen out during the fermentation process to prevent aerobic respiration in this investigation. 3.3 is again an AO3 question from the same topic of aerobic and anaerobic respiration. Suggest why the mixture was left for 20 minutes before the gas range was attached. The mixture of the sugar solution with the yeast both were left for 20 minutes at the temperature of 2 degrees Celsius. The reason is the mixture and the yeast cells both will get at equilibrium temperature of 2 degrees Celsius, so that the student can check the effect of temperature on the fermentation process. Let's see the model answer. The model answer is: the mixture and the yeast cells both reach at the same temperature of 2 degrees Celsius. Yeast cells and the mixture both will come to the temperature of 2 degrees Celsius. Step 1 to 4 of the method were repeated at 35 degrees Celsius. The volume of the gas collected was recorded every 5 minutes for 45 minutes. Table number 2 shows the results of two flaws for the first 30 minutes, whereas table number 3 shows the result of last 50 minutes when both the flaws are at 35 Celsius. If you're going to compare the tables, one flask was kept at 2 degrees Celsius and other flask was at 35 degrees Celsius. For last 15 minutes, these flaws were also moved to the temperature of 35 degrees Celsius, and we can see the volume of gas produced in both of the cases. 3.4 is an AO2 question. Explain the result from 0 minutes to 45 minutes of the flask that was at 2 degree and then moved to 35 degrees Celsius. Use table number 2 and table number 3. This question is again from aerobic and anaerobic respiration. If you look here, only this column, we can see that the flaws first at 2 degrees Celsius, we can see that there is no production of carbon dioxide gas. It means the fermentation process was not going on. But when the same flaws was moved from 2 degrees Celsius to 35 degrees Celsius, we can see the rate of gas production is increasing. At 2 degrees temperature, the temperature was so low, the enzymes in the yeast they were inactive to work, and there was no fermentation process going on. As a result, there was no carbon dioxide was formed in first 30 minutes. When the same flaws was moved from 2 degrees Celsius to 35 degrees Celsius, we can see a reasonable amount of formation of carbon dioxide. Let's see the model answer. The model answer says from table number 2, at 2 degrees Celsius the temperature was very low. The enzymes of the yeast are inactive to work. Therefore, no carbon dioxide is formed, meaning the fermentation process is very slow or does not occur. In table number 3, we are comparing the same flask, but when the same flask is moved from 2 degrees Celsius to 35 degrees Celsius in the water bath, the enzymes are active and a good amount of carbon dioxide is produced. 3.5 is a combination of AO1 and AO2 questions. Explain the results from 0 minutes to 45 minutes of the flask kept at 35 degree. Use table number 2 and table number 3. In 3.5, we need to compare the table number 2 and table number 3 for the flask which was kept at 35 degree only. We can see that the rate of production of carbon dioxide was quite good in the beginning, but later on the rate is getting slow here. The reason behind it is when the yeast start working, the glucose is converted into ethanol and carbon dioxide gas is formed. Initially, the quantity of the glucose was enough for the yeast to perform the fermentation process, but gradually the quantity of glucose is decreasing in the solution. Therefore, the rate of fermentation is slowing down and less carbon dioxide is formed because most of the glucose is used and the ethanol is formed. Let's see the model answer. In 3.5, we know that glucose is converted into ethanol and carbon dioxide with the help of the yeast in the fermentation process. In table number 2 and table number 3 are showing that the rate of fermentation is quite good at 35 degree and more carbon dioxide was formed. The rate of fermentation was very high for first 15 minutes, then it slowed down. The reason behind it is most of the glucose is used and the ethanol is formed. Question number 4. Pathogens are microorganisms that cause diseases. Gonorrhea, malaria, and measles are three different diseases in humans. 4.1 is an AO1 question and it is from communicable diseases. Draw one line from each disease to the pathogen that causes the disease. We have three different diseases: gonorrhea, malaria, and measles. The pathogen names are given here: bacterium, fungus, protist, and virus. Gonorrhea is a disease which is caused by bacterium. Malaria is a disease which is caused by protist. Whereas measles is a disease which is caused by virus. 4.2 is an AO2 question. Malaria is transmitted by mosquitoes. Male mosquitoes can be sterilized, so they are infertile. Here's a key information. The spread of malaria is reduced by releasing sterile mosquitoes into the environment. Explain how releasing sterile mosquitoes reduce the spread of malaria. By using the key information, by sterilizing, it means the mosquitoes are infertile. So they are unable to produce the offsprings. Therefore, less mosquitoes are in the environment which can bite the people. So less mosquitoes will pass the pathogens to the people. Let's see the model answer. The sterilized male mosquitoes are infertile. Therefore, less and no offsprings are produced in the environment. A fewer mosquitoes will bite the people, then less mosquito will pass a pathogen to the people. Therefore, the transmission of malaria can be controlled. Pathogens also cause diseases in plants. Figure number 4 shows a rose black spot, fungal spore, and a tobacco mosaic virus are shown in figure number 4. In figure number 4, rose black spot fungal spores are given here, which are 16 micrometer, whereas tobacco mosaic virus, the size is given 2.5 10 raised power minus 7 meter is there. Images are not in the same scale. The reason is micromean 10 raised power minus 6 meter. 4.3 question says that name the piece of equipment used to view the virus there. It is an AO2 question, and this question is from microscopic topic. To view the virus, we can't use the light microscope. We need to use the electron microscope to view the virus, which is given in figure number 4. The model answer is electron microscope. 4.4 is an AO2 question from the topic of microscopy and communicable diseases. How many times longer is the fungal spore than the virus? We can use the figure number 4. The size of the fungal spore is 16 micrometer. Micromean 10 raised power minus 6 meter. The size of the virus is also given, that is 2.5 10 raised power minus 7 meter. We want to see that is the fungal spore is larger than the virus, then we see the ratio between them. If the answer is greater than 1, then the fungal spore is larger than the virus. So we took the ratio size of fungal spore to size of the virus. Size of the fungal spore is 16 into 10 raised power minus 6 meters, whereas the size of the virus, which is given in figure number 4, is 2.5 10 raised power minus 7. The student need to use a calculator and the answer is 64. This means that the fungal spore is 64 times larger or longer than the virus. 4.5 is a combination of year 1 and year 2 questions, and this question is from the viral diseases and photosynthesis and respiration. Explain why plants infected by tobacco mosaic virus grow slowly. Tobacco mosaic virus destroy the chlorophyll in the leaves. It affects the quantity of chlorophyll in the leaf. Therefore, the plants are unable to do enough photosynthesis to increase the quantity of glucose. Let's see the model answer. The model answer says tobacco mosaic virus destroys the chlorophyll in the leaves of a plant. As chlorophyll is affected, therefore the plant is unable to do proper photosynthesis. Therefore, less glucose is formed for other chemical processes such as amino acid, cellulose formation. Less energy is produced for other chemical reactions. Question number 5. Question number 5 shows the human heart. This is the structure of the human heart. The human heart consists of two parts. One is the left side of the heart, which is related to the oxygenated blood, whereas the right side of the heart is related to deoxygenated blood. The heart consists of left ventricle, left atrium, pulmonary vein, and aorta, which is carrying the oxygenated blood to the body. Whereas the blue area of the heart is showing the right ventricle, right atrium, deoxygenated blood is coming from the body, and this is pulmonary artery taking the deoxygenated blood towards the lungs. Let's see the first part of the question. 5.1 is an AO2 question, and this question is from the heart and blood vessels. Which blood vessel transports blood with the highest oxygen concentration into the heart? So the question is, among these four, A, B, C, D, which chamber or which artery or which vein is responsible to bringing the oxygenated blood into the heart? If you see here, the blood is entering into the heart. That is, pulmonary vein is the vein which is carrying the oxygenated blood from the lungs into the heart. So it is number D. 5.2 is an AO2 question, and this question is again from the same topic, the heart and blood vessels. Blood pressure is a measure of the force of the blood against the walls of the blood vessels. Which blood vessels transport blood at the highest pressure? Mean which will pump out the blood at the high pressure. If you look here, we have A, B, C, and D. When the blood is pumped from the left ventricle into the aorta, the blood has a high pressure. because the blood is going to the rest of the part of the body. Therefore, the blood in this vessel has the highest blood pressure. So our answer is number C. 5.3 is an AO1 question. What is the correct order for the blood flowing through the heart to the lungs? So it means that we have to see from our figure number 5 which vessel is taking the blood to the lungs. We see here the blood is coming from here through the pulmonary artery, it is going towards the lungs. Whereas aorta is responsible to carry the blood to the rest of the part of the body. So order is the right atrium, the right ventricle, and towards the pulmonary artery. So this is the correct option. Every year, thousands of people in the United Kingdom have heart attacks. A heart attack is caused when the heart muscle cells do not get enough oxygen, causing the cells to die. 5.4 is an EO3 question. Statins and stents are two treatments used to reduce the risk of someone having the heart attack. Evaluate the use of statins compared with the use of stents to reduce the risk of the heart attack. This question is from coronary heart diseases, and student must have the information what are statins and what are stents. Statins are actually the drugs which are used by the people to minimize or lower down the level of cholesterol in the body. High cholesterol level can increase the fats in the arteries, which can lead towards the heart attack. Whereas stents are the small tubes which are injected in the arteries to open the arteries for the blood flow. Here are the advantages of given, and we can compare the advantages of each other. The statins and stents. Statins are easy to use. Statins can help to reduce the cholesterol level in the body, which reduces the risk of heart attack or the stroke by minimizing the level of cholesterol in the body. Statins slow down the build-up of the fats in the arteries. Now we are going to see the advantages of the stents. Stents helps to open the blocked arteries. They help the flow of the blood through the heart muscles easily, and it increase the flow of the blood. Statins remain in their places for longer time. Now we are going to compare the disadvantages of both of them. Statins are the drug, and drugs will need to be taken for a longer time on regular basis. Statins take the time to show its effect. Statins might have the effects like muscle pain or kidney failure. If the people forget to take the statins, the level of cholesterol will start increasing in the body. Now we are going to see for the stents. As surgery is required for stents in the artery, therefore there is a chance of the heart attack due to the bleeding. Blood can clot in the artery near the stents, and it can block the arteries again. There are risks of infection from the surgery. So from this question we saw there are advantages and disadvantages of statins and stents. The student needs to write the three advantages by comparing and three disadvantages for comparing statins with the stents. They shouldn't write only about the statins or stents only. Otherwise, they will unable to score 6 marks. 5.5 is an AO2 question, and this question is from blood vessels, coronary heart diseases, and respiration. Many people who survive a heart attack get out of breath easily when they do exercise gently. Explain why heart attack survivors get out of breath easily. Due to the heart attack, their heart muscles get weaker, so they are unable to perform well and they are unable to take the proper oxygen for respiration. If they take less oxygen, so they will produce less energy for heart cells or tissues. Therefore, the person who had heart attack will get out of the breath easily during the light exercise. Let's see the model answer. The model answer is the heart muscles get weak after heart attack. Therefore, heart is unable to pump the blood properly throughout the body. The body will get less blood from the heart. The body will get less oxygen for respiration. Less oxygen means less energy supply is there. Therefore, breathing rate will increase to supply for more oxygen to the body. Scientists have developed patches of beating heart cells to repair damaged heart tissues. The patches are placed onto the area of the heart where the cells have died. New cells grow to replace the dead cells. The patches are made by using the man's own cells. They are converted into stem cells. 5.6 explain why stem cells are used to make the patches. It is a combination of A1, A2 question, and this question is from coronary heart diseases and stem cells. Now the questions come here, why the stem cells are used. Stem cells are actually undifferentiated or unspecialized cells, so they can develop into any kind of the cell there. They can develop into the muscles, they can develop into the heart cells, therefore the stem cells are used. So these patches are used from the person's own cells and they are converted into the stem cells. The advantage is they can convert into any kind of the cell in the body. Let's see the model answer. The model answer is stem cells are undifferentiated cells, mean unspecialized cells which can become any type of the cell that is needed. Therefore, these cells can be developed into heart cells or the muscles. 5.7 is an AO3 question. The scientist could have used human embryonic stem cells to make the patches. Give two advantages of using stem cells made from the person's own cells rather than using embryonic stem cells. We have seen from the previous question that the patches are made from the person's own cells instead of taking from embryonic stem cells. The advantage of doing this thing is these new stem cells are not rejected by own body. The reason is these cells are taken from the person's own body. So these patches are not affected by human immune system. The second thing is there is no point to destroy the human life, means the human embryo. On the other hand, an adult person or adult human can give consent whereas an embryo cannot do that. Let's see the model answer. Stem cells are not rejected by own body because the patches are made from the person's own cells. There is no point to destroy the human life, means to damage a human embryo, whereas an adult human can give consent but an embryo cannot. Question number 6. The question is about plant transport system. 6.1 is an year 1 question. And this question is from the topic of osmosis, plants organs, tissues and organ system. Describe how water is transported from the soil to the atmosphere through a plant. The plant has root hairs, and the root hairs absorb the water from the soil through the osmosis process. From there, the water is taken to the leaves through the hollow tubes, which are called xylem vessels or xylem tissues. The plant uses that water during photosynthesis, and the rest of the water is released into the air through tiny holes, which are called stomata, through the transpiration stream. The model answer is plants' root hair cell absorb water from the soil by osmosis process. The water travels from the root to the leaves through the xylem vessels or tissues. The water will lose through a lot of tiny holes which are called stomata on the lower side of the leaf which are present through the transpiration stream. 6.2 is an AO1 question, and this question is from the plant organ system. Dissolved sugars are moved through the plant in flowing tissues or flowing vessels. What is the name of the process that moves dissolved sugar through phloem tissues? So it is an year 1 question. Student must remember the name of the process which is carried out by the phloem tissues to transport the food, that is called translocation. Is one more question and it is a keyword. Student must be careful about the spellings. Figure number 6 shows the structure of a phloem tissue. A phloem tissue consists of two cells. One is called the sieve tube cells, another is the companion cells. Phloem tissue is a specialized cell. It carries the food across the plant. And this topic is from specialized cells, plant cells and organs, and plant organ system. The C plates have a lot of holes present here so that the dissolved sugar can move up and down throughout the sieve tube in different parts of the plant. Now we are going to see the question. 6.3 is an AO1 and AO2 question, and it is 2 mark question. Explain one way sieve tube cells are specialized for their function. Use figure number 6. Sieve plate cells between the cells have lot of pores so that the sugar solution can easily flow up and down between the sieve tube cells. Let's see the model answer. The sieve plate between the cell have the pores which are given here, you can see in the figure, so that the sugar solution can easily flow up and down between the sieve tube cells. 6.4 is a combination of AO3 and AO2 question, and this question is from cell specialization, plant cells and organs, and respiration. What does the structure of the companion cells suggest about the process that moves dissolved sugar through the phloem tissues? Give a reason for your answer. Use the figure number 6. If we go back to figure number 6, from figure number 6 we can see this is a companion cell and there is a large number of mitochondria present here. The mitochondria's job is to produce energy, and the sieve tube cells they need energy. We can see that they are hollow tubes, but these cells need energy. So mitochondria will produce energy, the companion cell will use energy, and the extra energy is given to sieve tube cells so that they can work and move the sugar up and down in the plant. Let's see the model answer. The companion cells have a lot of mitochondria. They will provide the energy to the sieve tube cells so that the dissolved food can move up and down the plant in the phloem tubes. 6.5 is a combination of AO1 and AO2 question. Describe why it is important that dissolved sugars are moved both upwards and downwards in a plant. This question is from the plant cells and organs, organ systems, metabolism, use of glucose from photosynthesis. During photosynthesis, the plant is making glucose, a sugar, and all the living cells in the plant they need energy. Therefore, the phloem cells are phloem tubes. They help to transport the food to every single cell of the plant. Plant cell needs sugar for respiration, for their growth and cell division, and the extra sugar is stored in the form of starch or fats or oil in the plant. Let's see the model answer. The model answer is sugar is made in the leaves during photosynthesis. Student can use the word glucose instead of sugar. All living cells or tissues need sugar for respiration, for the growth and cell division. The extra sugar is stored as starch, fat, or oil in the plant. Therefore, the dissolved sugar is moved both upwards and downwards in a plant.